Okay guys, so in this lecture we are going to talk about the trip operon. Trip operon, like the lac operon, is also a bacterial operon present in the E. coli, and it has genes which are responsible for coding for those enzymes or enzyme subunits that are involved in the biosynthesis of tryptophan amino acid. Although tryptophan is an amino acid, but its synthesis involves several enzymes. And although these enzymes are much larger molecules, they contain several amino acids themselves, but they are involved in the synthesis of tryptophan amino acid. Now, let's look at the structure of trip operon. I told you what operon is in the previous lectures as well, the link to one of which has been given in the description. So do check that out if you don't know what operon is. Okay, so now basically an operon, trip operon. Trip is from tryptophan amino acid, and operon basically that operon, that functional unit of DNA that controls or that regulates the genes which that contains the, I should rather say, the genes which are involved in the biosynthesis, in the production of those enzyme subunits or enzymes themselves that are involved in the biosynthesis of tryptophan. Now, this operon, also like the lac operon which we did in the previous lecture, contains a promoter region, an operator region, and five structural genes. The operator region and promoter region, they partially overlap with one another, and this is its entire structure. Now, present in the E. coli bacterium. Tryptophan molecule is an amino acid. Okay, so once the operator is empty and the promoter over here is at the promoter region, the RNA polymerase can come and bind. And once the RNA polymerase binds and the operator is empty, so it will initiate the transcription of these structural genes. Okay, the names of these genes are trip E, trip D, trip C, trip B, and trip A. Now, and after these enzymes are formed, so these enzymes through several steps will synthesize the tryptophan amino acid. Now, tryptophan molecule itself, it helps as a regulator. It helps in regulating the expression of the trip operon. How? Well, it acts as a core repressor. It acts as a core repressor, okay, and it helps in the repression of the trip operon and binds to a repressor molecule, which is somewhere in the cytoplasm of the E. coli. Repressor molecule, after the repressor molecule binds to the tryptophan molecule, they form a complete repressor, and then that active repressor will go and bind to the operator side, and then after binding to the operator side, the activity of RNA polymerase will be blocked, and so transcription will not be initiated if tryptophan is present. This is very important because obviously if tryptophan is present, there is no need to make more tryptophan. There is no need for wasting more energy in synthesizing these enzymes and then producing more tryptophan. Only these enzymes should be made if tryptophan is lacking or scarce. So if tryptophan already is present, it will act as a co-repressor, and then it will bind to a repressor molecule in the cytoplasm. After binding to the repressor molecule, the complete activated repressor will go and bind to the operator region. And after binding to the operator region, although if the RNA polymerase comes and binds to the promoter region, it will not be allowed to initiate this transcription process because the operator side, the repressor at the operator side will block its activity. Okay, so this is one way how the tryptophan regulates the expression of the trip operon. And if tryptophan is scarce, obviously, so then it will not be able to bind to the repressor molecule and will not be able to activate the repressor, and so this operator site will become empty. And if this is empty, then the RNA polymerase will come and bind to the promoter region and then initiate the transcription of these genes, these structural genes. Now, let's go over the important points that I've listed over here. In addition to tryptophan acting as a co-repressor, it helps in regulating the trip operon through the process of attenuation. Okay, guys, now this is another mechanism by which this trip operon is regulated, and again a very important mechanism, because the tryptophan itself, by acting as a co-repressor, is not sufficient in repressing this trip operon. Like, it's sometimes it's incompletely repressed, it's not completely repressed, so this other mechanism, the attenuation, is needed. Attenuation literally means the immature termination of the mRNA synthesis. Okay, it immaturely terminates the transcription. It means a premature or immature premature termination of the transcription process by forming an attenuator. Attenuator is a hairpin-like structure which is formed by the self-complementary regions on the mRNA molecule. Self-complementary sequences are made in certain regions of the newly formed mRNA, and they pair up to form a hairpin-like structure. And this hairpin-like structure, it attenuates the transcription process. It actually stops the transcription process. So this is the second mechanism by which it is regulated. So these are the two main mechanisms by which the tryptophan operon, or the trip operon, is regulated, and both of them are through negative regulation, because both of these mechanisms, they actually tend to decrease the expression of the trip operon. So this is and again an example of negative regulation, and we've talked about the definition of negative regulation in the last lectures as well. So I'll try giving the link to that as well in the in the description. Now, in prokaryotes, okay, uh, if tryptophan is not available, okay, if tryptophan is available, so what will happen? This attenuator will form. But in prokaryotes, if tryptophan is not available, then the bacterium needs to make more tryptophan, more enzymes, so that more tryptophan can be synthesized. So in that case, what happens? For in order to understand that, we must know the fact that in prokaryotes, the transcription and the translation processes, the formation of the mRNA and then translation of that mRNA into a protein, they occur side by side. They are linked to one another. Okay, so the translation of mRNA, it actually begins even before the transcription of mRNA is fully completed. So after knowing this fact, at the 5' end of the mRNA, there are two codons that code for tryptophan itself, the tryptophan amino acid itself. So if tryptophan is available, then the translation continues across these codons as well, because now tryptophan will come and bind to these codons, and the ribosome will be able to form to continue the translation. But if tryptophan is not available, then what happens? If tryptophan is not available, so think about it, we'll need more tryptophan. The bacterium needs to make more tryptophan. So in order to do that, if tryptophan is not available, these codons, which I just told you that there are codons near the 5' end, these codons will not be able to fill. They'll remain empty, and if they'll remain empty, so when the ribosome reaches these codons, it will stop, because there is no tryptophan to attach at these codons, and it will wait for the tryptophan molecules to come. Once it stops, what happens? That the ribosome, it kind of covers the attenuator sequences. It covers those codons which are necessary for the formation of the attenuator, for the formation of that hairpin. So if the ribosome covers the hairpin or the attenuator sequence and does not move forward, so what would happen? Transcription of the mRNA molecule will still continue. And so if transcription continues, so then later on, the mRNA molecule will be translated into enzymes that will be responsible for the formation of tryptophan. So these are actually the two processes through which tryptophan metabolism is controlled. A tryptophan. Therefore, so if tryptophan is scarce, the attenuator is not made, and hence trip is expressed. Okay, so if and obviously because these two codons are empty and the ribosome comes and it covers attenuator sequences, and this forms more tryptophan. Okay, I took a diagram from your book, Illustrated Reviews of Lippincott. In here, we can see the ribosome right over here. It is translating this synthesizing mRNA molecule, and this is the hairpin-like structure that I was talking about. This hairpin-like structure is the attenuator sequence, and now, before if the what you call if the tryptophan molecules are available, then the ribosome, it will pass over the attenuator sequences and will not cover the attenuator sequences. It will not cover the attenuating, it will not cover the attenuators codons, and hence attenuation will occur. But if tryptophan is not available, then the ribosomes, it will cover those codons that are involved in the formation of the attenuator. And so if it covers the codons that are involved in the formation of the attenuator in that case will not form, and this mRNA synthesis will continue and elongating the mRNA chain. And so tryptophan biosynthesis can occur by if that mRNA molecule which has been synthesized and which has been synthesized uh, because the mRNA molecule can then be translated into those enzymes which are involved in the biosynthesis of tryptophan. Now I'll just read over this part. Self-complementary sequences in certain regions of the mRNA causes a hairpin to form, which attenuates prematurely. And transcription, we've talked about this, that these self-complementary sequences, they form this attenuator. Okay, they basically they pair with one another and they form the attenuator sequence, the hairpin. And ribosomes, they initiate translation at the mRNA as the mRNA is being transcribed. And we've talked about this part as well because they are actually linked in prokaryotes. The transcription and translation are linked. The translation of the mRNA chain, it starts out even before the translation of the mRNA molecule starts out even before the transcription of the mRNA is complete. Okay, so the two ways: if we have abundant tryptophan, it will act as a co-repressor and it will bind to the repressor, it will activate the repressor, and then this repressor will go and bind to the operator region. And after binding to the operator site, it will block the activity of RNA polymerase. So this is the first role of tryptophan. And then the second role of tryptophan is that it allows the formation of mRNA hairpin. It allows the formation of hairpin attenuator, because if it is present, it will be available for those two codons that we just talked about at near the 5' end. They'll be available to bind and to come and bind at those two codons, and so the ribosome will not cover the attenuator sequence and will allow the attenuator to form, and therefore prematurely the mRNA molecule will be terminated. The mRNA synthesis will be terminated in that case. Okay, so that's it. Thank you so much. Next lecture coming soon.